Okay, so our last little bit for cells for this week is going to be about the cytoskeleton. Get my screen up here, get this into slideshow mode. Okay. Again, like I usually do, here's a link to the section of the textbook so that you can have that available to you, really handy dandy. There is one more section in this chapter, which is cell communication, which I'll pick up either next week or I'll have it be, I'm going to be gone next week. So we might have that be the Thursday activity instead of it being something that we do in class. Okay, I'll figure it out. But for now, this is going to be our last section for what we're going to be doing on Thursday with the cytoskeleton. Okay, let's get into it. So what the heck is a cytoskeleton? What are microfilaments, intermediate filaments, and microtubules? What are cilia and flagella? How are they similar and different? And then we see the differences between prokaryotic animal and plant cells and how their cytoskeleton is arranged. Okay, so what is a cytoskeleton? Well, basically it is a network of protein fibers that secure organelles into specific positions. It provides structure, it determines cell shape, and it gives the cell strength so it doesn't get smashed really easily. It allows the cytoplasm and vesicle movement inside the cell, and it allows us multicellular. We can move around because of our cytoskeletons. And so we're going to go through these different fibers here. And so I've arranged them here from the small to big, but then when we talk about them, we're going to go big to small. So sorry about that. But we have our microfilaments, which are also called actin filaments. And these are just proteins that are just kind of twisted around each other. So if you think of two ropes that are just kind of twisted around like that, that's what these actin filaments are. And then we've got intermediate filaments. These kind of have a different structure. The way that they're put together and broken apart is different from the other two structures. And they're kind of like in between sizes. So we call them the intermediate ones because they're not big and they're not little. So we'll just call them intermediate. And then we have our microtubule. And this is a potential point of confusion that you need to be careful for because you're like, ooh, yeah, micro, that means small. But tubes are much bigger than filaments. Filaments are very small, hair-like things, or tubes are like pipe-like things. So just if you can kind of think about the different, so if you can think of filaments as being hair-like, these are tiny hair-like things, and then microtubules are little tiny pipes in our cells, but microtubules are bigger than filaments. So kind of just make sure that you can get those concepts and be able to arrange these from big to small. Okay. So let's start with microtubules. These are our biggest ones. They're about 25 micrometers, which is, you know, still pretty little, but it's the biggest one of our cytoskeleton components. These are made of alpha and beta tubulin, which is just fancy proteins. And so these form a long hollow tube that's a big long cylinder. So it's literally like a pipe. And then there's a positive end and a negative end on these guys. And proteins are added to the positive end to make this grow bigger. And then the negative end is where it's broken down and it grows smaller. And so we can actually see how we might start here. So here's a positive, we're going to add to it, and then we're going to take some away, and we're going to add to it and take some away. And you can see how this tubule can move around the cell based on how it's added to and taken away from. And this has this dynamic instability thing where it's constantly growing and shrinking, and it's getting broken down and built up. And this is important for how chromosomes are sorted out during cell division. And then I encourage you to watch this little 30-second, it's like 39 seconds or something, YouTube video showing what this looks like actually under a microscope. So definitely check that link out, because it's worth the 30 seconds of your life to understand this. It's pretty, and it's kind of pretty. So that's pretty dope. Okay, so what do microtubules do? So here's a link to a little GIF of a little paramecium type single cellular thing changing shape. And so you can link to that GIF there. But I put on here, you know, we have muscle cells and blood cells and bone cells and reproduction cells and fat cells and nerve cells, and all of these have different shapes. And you got to remember, inside your body, you have all these different types of cells, but they all have the same genes. And the reason that they can do the different jobs that they do is because of how they are shaped. And so we have genes turning on and genes turning off, and genes only turned on a little bit, genes turned on a lot. And that tells our cells how to differentiate and how to grow up to do the jobs that they need to be done in the cell. And so one of those, form follows function, right? So we have, in order for a muscle cell to do a muscle cell's job, it has to have that shape. And it's these microtubules and the other components of the cytoskeleton that make the cells that shape. So the microtubules also help organize the organelles, so they make sure that everybody's in the right place, doing the right thing. Again, that as our cells are dividing, we'll go through the different steps of meiosis, but when those cells are dividing, we got to make sure that we have the genes, the chromosomes, in the right places. So when those cells pinch off, there's DNA in both cells. And that's one of the things that the microtubules do, is they make sure that those chromosomes get to the right place at the right time during cell division. And then we have cell mobility that's controlled by cilia and flagella. So cilia, you can see from this image here, these are a bunch of little tiny hairs that completely cover the cell, and they kind of like wiggle around, and they create these waves that allow the cell to be propelled through this watery substance. Flagella are usually fewer, and they have longer whip-like tails. But the cilia and the flagella are both controlled by microtubules. Okay, so that's their jam. And you guys can see these little GIFs that are, or GIFs, I don't know how you say it. People have a lot of feelings about how that's supposed to be pronounced. But anyway, so you can link to those just to kind of play around and see those. It's kind of fun to see little cells swimming through water. All right. And then, so now we're going to switch to the intermediate filaments. These are about 10 micrometers or microns. So these are less than half of the width of the guys that we just talked about. Not all animal species have these things, but most of them do. And they kind of form this staggered twisting rope thing. So if you've ever unwound a rope, you know that it's just a series of little tiny strings being twisted around and around and around and around and around, and then increasing the diameter. So you start with little tiny threads, and then you get bigger cords, and then you get a rope, and then you get, you know, so that's basically what is happening here in these intermediate filaments. And what they do is that they create some tension, and that tension stabilizes the shapes cell, the cell, nope, it stabilizes the shape of the cell. That was really hard to say. And it provides its structure. So just like a tension bridge is held together by things pulling against each other, that's the same thing happening here that allows our cells to be that shape. So here's one of those things where they're not where they're unlike the other two kinds of cytoskeletal filaments is that these don't really have that dynamic instability where they're growing and shrinking and growing and shrinking and growing and shrinking. Once they're built, that's kind of how they stay. They don't really change length as much as the other guys do. And then they're made of, you know, different kinds of proteins depending on what their jobs are. They can be keratin, and they could be this other thing called lamin. It could be other things that we're not going to list, but you know, there's a bunch of stuff. So again, just like all the cytoskeletal filaments, they help provide shape for this cell. And this one really provides mechanical strength. And so if you know, when I do that to my hand, it affects my cells, but they're able to withstand that pressure because of these intermediate filaments. And they also provide anchor points for the plasma membrane and the nuclear envelopes. And so that those structures, the plasma membrane and the nuclear membrane, can maintain their shapes as well. Okay. And then our last and smallest are the microfilaments. So these are the teeny hairs. This is the skinniest of the three protein fibers that make up the cytoskeleton. They're only 7 microns, so they're a little bit smaller than the intermediates. And these are 2 globular proteins that are twisted around each other. So if you think of it, just kind of a nice little twist like that, that's all they're doing. These little skinny strands are just twisting around each other. We see these concentrated near the plasma membrane. And so if we look at this diagram that I threw in here, this little kind of bluey purple thing that's near the outside of the cell, those are the microfilaments, which are also called the actin filaments. Actin is the protein that makes these things up. So these are really to hold that plasma membrane in place. And then we can see where the other ones are at the other. So then we have the intermediate filaments kind of on the inside of those actin filaments. And then we have the microtubules that are concentrated near the nucleus. So, okay, so let's go back. Let's go back and talk about actin filaments and not get sidetracked like I am prone to do. So these are the skinny ones. The actin proteins are wound up together, concentrated in the plasma membrane. And these also have dynamic stability where we have a positive end. Let me grab we have a positive end and a negative end. And so the positive ends are where the growth occurs. And then the negative end is where it's broken down. Positive end is growing. Negative end is the breaking down. Okay. So again, microfilaments, just like all of our cytoskeleton proteins, they affect shell shape and the actin filaments do this by supporting the plasma membrane. They provide strength for the cell, and they move organelles and vesicles, all that stuff moving around inside the cells. And again, these are important for cell division in animals. Plants, because they have that cell wall, they're able to move those chromosomes using a different mechanism. Okay, and then the last thing that I wanted to talk about are motor proteins. So these are pretty cool. So we talked a little bit before about flagella. It's this long whip-like tail that allows cells to move through aqueous environments, or they might have cilia, which are these the shorter hairs that are completely cover the cell, and they kind of swim around, and they're really cute. So definitely check out those little GIFs or GIFs. But these are controlled by motor proteins. And these motor proteins have a 9 and 2 arrangement. And so like if we look at this micrograph here, we can see that there are these proteins that are kind of arranged in couplets, right? There's 2 of them, they're kind of form one unit. And if we counted those 1, 2, 3, 4, 5, there'd be 9 on the exterior and then 2 in the middle. And it's how the relativeness of how those these proteins are triggered that allows this whip-like action of the flagella that propels the cell through the aqueous environment, the watery substance that it's swimming through. And so here, this is just a figure that's kind of demonstrating in the principal band, you know, we've got these guys that are activated, and then in the straight region, you know, everybody's kind of chill. And then in the reverse bend, it's these guys over here that are activated. And so it's just the point of this is it's that cytoskeleton that controls which of these protein couplets are being moved, and that's what controls the motion of the flagella or the cilia that allow the cell to move through the watery substance. Okay, so that is all that I have for cells. So on Thursday, what we'll do is I'm starting to come up with some ideas of how we can actually practice this and use some manipulation instead of me just blah, blah, blahing at you and then you trying to have to remember it later. We'll do some kind of hands-on movement of stuff, trying to do some puzzles and stuff to figure that out. But let me know if you have any questions. And I will see you guys on Thursday. So yep, and we'll talk more about what the next week is going to look like on Thursday. Okay, talk to you guys later.